Each cell goes through the process of cell cycle, which is consisted of interphase, mitosis, and cytokinesis. Cells go through the cell cycle by the signaling of neighboring cells. They replicate another exact copy of them to replace the cell that is damaged or dead. In our human bodies, 300 million cells are replaced every minute. For a cell to go through the complete cell cycle, it takes about 24 hours, most of it spent in interphase. Interphase is called so because it is the period in between one cell division and the next. Therefore, it is the phase in which a cell gets prepared for cell division. Interphase can be divided into gap 1, synthesis, and gap 2 phases. Gap 1 phase is the first period of growth, in which the cell does most of its growing. The cell increases in size about 20% at the end, and also synthesizes new proteins and organelles. As our synthesis phase is the phase in which a cell synthesizes the necessary organelles and replicate the DNA and centriole, which are needed in the cell division. At the end of the synthesis phase, the cell has twice the amount of DNA it had at the beginning of the cell cycle. After synthesis phase, cell goes to the gap 2 phase, which is the last phase of the interphase and the last period of growth. During the gap 2 phase, cell grows a bit more in size and produces the organelles and molecules needed for the cell division, which is the next step. Mitosis can be divided into 4 phases, which are prophase, metaphase, anaphase, and telophase. During prophase, the genetic material inside the nucleus or the nucleus envelope condenses and takes the form of the classic structure of X-shaped duplicated chromosomes. Its DNA strand of the chromosomes is known as chromatid, and they are attached together by the area of centromeres. The spindle fibers, which is a system of microtubules, start to extend from the centrioles outside the nucleus. Eventually, the nucleolus and nuclear envelope break down at the end of the prophase, with spindle fibers extended and DNA molecules condensed into duplicated chromosomes. The spindle fibers from the prophase connect centromeres of the chromosomes to both centrioles at each end of the cell during metaphase, as the chromosomes line up at the center of the cell. Anaphase begins as the sister chromatids are separated into 2 individual chromosomes and move to each end of the cell towards the centrioles along with the spindle fibers. Anaphase is followed by the last stage of mitosis, which is telophase. During telophase, the formed condensed form of chromosomes go back to the loose, tangled form of chromatin, and new nucleus and nucleus envelopes are also produced, one at each end of the cell. During the last stage of the cell cycle, cytokinesis, the cytoplasm are divided in 2. Thus, 2 daughter cells are produced, which are the outcome of the cell cycle. However, how cancer is developed in your body is related to the cell cycle. The cell cycle is regulated by the balance between 2 types of genes, proto-oncogenes and tumor suppressor genes. Proto-oncogenes promote and accelerate cell division, while tumor suppressor genes inhibit cell division. Tumor and cancer-causing cells are those who follow their own pattern of cell division, having a tendency to divide more frequently due to the changes in DNA. The changes in DNA result in making proto-oncogenes become oncogenes, accelerating cell division even more, where tumor suppressor genes lose the power to suppress the cell division. They cause tumor, which displaces and kills normal cells nearby. Chemical poisons, family history of cancer, radiation exposure, and UV light are the factors that may cause cancer because they result in causing mutations, which cause mutations or changes in DNA and make the 2 genes imbalanced.